Welcome. We are excited to hear from our next group of presenters. In the first session, we'll hear from Dr. Matt Giel and learn more about the role of the Global Research Network. Some of you may be familiar with the GRN, and for others, this will be an opportunity to hear from the dedicated team that focuses on PURA from a research perspective. We will then show a pre-recorded presentation from Dr. Dirk Niessing on how to read a genetic diagnosis. Dirk could not be with us in person today, but will be with us at the final element session on Saturday, June 18 at 11 a.m. Following Dirk's remarks, we are pleased to have Sabrina Bakker, Sandra Burchick, and Carolyn Kettler, PhD students at the Niessing Lab, to present on antibodies against PURA, PURB, making the invisible visible. It's an amazing group, and we are so fortunate to have them all with us. Let's get started. If you would like to ask a question, please use the question box located at the bottom of your screen. We'll have time for questions after this session, and remember that you may submit questions at any time on the conference website for any of the research topics. We will have a final element session on our last day, Saturday, June 18, devoted completely to your questions. There is a lot of information to process, and you may think of something through the week that you wanted to ask. And now, please welcome Dr. Matt Giel. Matt grew up on the tiny island of Guernsey before obtaining his degree and PhD in biochemistry at King's College London. After 3 years of postdoctoral work in cell structure at what is now the Crick Institute, Matt returned to working on gene regulation in the frog Xenopus, his PhD subject. At the University of London's Developmental Biology Research Center, he was part of the team that discovered how GATA transcription factors control cardiovascular system formation. Matt then established his own lab, continuing these studies at Portsmouth on the south coast of England until he was asked by the Xenopus research community to create a resource center for genetics in the model. Work on this began in 2006, and the EXRC is now the largest center for such studies, with visitors coming to train and do experiments from all over the world. With the advent of gene editing, which works extremely well in the frog, Matt sought collaborations with clinical geneticists to test whether the frog can be a useful tool to help them diagnose and understand rare genetic diseases. Among the diseases he and his colleagues have discovered and or investigated with Sarah Ennis and Diana Barali at Southampton is PURA. In his spare time, Matt coaches on Britain's Olympic rifle pathway. Welcome, Dr. Matt Giel.
Thank you very much, Damon. That's really kind. And I'll start by sharing my screen, I hope. Let's see. That's the one that I want. And I'm hoping that you can see the last slide. You'll get a very rapid backwards. There we go from this. So as Damon said, my name's Matt Giel. I'm a geneticist, and I actually have, I think, the world's largest frog collection. But here I've taken over from Dirk Niessing, who's been coordinating the Global Research Network for several years now. And I have to say, has been an absolute star in this role. And I, for one, really thank him for that. So we've, within the Global Research Network, we've tried to spread the workload a little because Dirk was doing so much of it. So a small group of us will be making sure that the network functions well. Really, the network is here around discovering the cause of PURA. We know that the gene change is a cause, but how does that actually give the cell and the person the problems that they do? Because if we can discover that, then in this lovely quote, there is hope for a cure. And I think you'll be hearing later for some lovely work on Rett syndrome that says that when we do understand at the molecular level the cause for a disease, maybe we're a little closer, even on rare genetic diseases, which are going to be extremely difficult for us to work with. Maybe we're starting to get pathways and ideas how in the future, and I'm afraid it's likely to be some time in the future, there might be ways to mitigate and this damage to the cell and maybe get towards a cure. So I think that's an exciting talk later on today on Rett syndrome. So I'm going to talk a little bit about the research interactions around the GRN, but also I wanted to just put in here a little bit about the way scientists work. Now, I think some of you who've dealt with the GRN over years are probably very familiar with that, but I'm thinking that also there's going to be some people who are new to Pura, and it'll be helpful, I think, for you to understand the way that scientists work. Then a little bit about what we know, what we knew about Pura function, a lot of it even before we knew that changes to the Pura gene and protein caused a disease. And then talk a bit about what we're going to do to find out more, or what we're doing to find out more, and around the key questions that we're asking as scientists, particularly the discovery scientists who work on Pura. So asking questions about what's the effect on isolated cells of changing the Pura protein or taking away the Pura protein. What's the effect in a more complicated mixture of cells? And we do that in model animals. And then finally, trying to understand what seems to be quite a recalcitrant question. How does Pura structure, the protein structure, and the function relate to one another? Because one of the unusual things perhaps about Pura is that you seem to be able to have variants of the Pura protein in patients that are quite widely different from one another, but cause very similar illness. To do that, I'll talk a little bit about the experimental tools that we use. And then right at the, well, towards the end, there's some very exciting data just about to come out from Dirk's lab that might start to be providing the very first clue about the answers to the molecular mechanism of Pura in the cell. They're very exciting. Still a long way from providing clinically useful knowledge, but certainly moving us in the right direction about understanding Pura. So that's very exciting. I was excited to read that. So let's start off with where the science fits, and let's look at this. So I think that that almost everyone who's involved in discovery research, so very much laboratory-based research on Pura, also interacts with clinicians, clinical geneticists, who are in turn involved in analyzing large numbers of rare genetic diseases and have come across Pura in their work. Then that sort of research group overlapping and working together provide both information, just like we are today, to you and via your doctors, and also, I think, tools. In time, we will have more tools in terms of genetic analysis, in terms of perhaps even being able to use the antibodies you'll hear about later today to understand more about an individual's issues, if you like, with the Pura protein. And again, coming through your doctor to you and your family support. But that information doesn't just flow one way, it's a two-way flow. Of course, we need more and more information from you directly as patients or your supporting family and your doctor. And also, it sounds awful to call this a tool, but I couldn't think of a better word, things like biobank samples. These are tools which we as researchers can then use to learn more and more about Pura. So this is all a very important role of the Pura Syndrome Foundation, which is to really bring these groups together so that we can get the very best information, tools moving across our whole group, so that in the end, patients may be able to be better one day. I have to say that there was a bit of a disappointment for me stepping in as the coordinator for the first time, that when I contacted a lot of people, there has not been an enormous amount of progress in the last 12 months or so, because actually the COVID pandemic slowed things down, not only in labs, but I know, for an example, in my own lab, normally we can get DNA sequenced in 2 days. At the moment, it's taking more like 2 weeks. And so the COVID pandemic is still causing things to be slow. And so research is not moving as fast as it normally does. It's getting back to normal, which is the good news, but it has had an effect. So I'm kind of a little bit disappointed not to be able to tell you more, but very excited to be able to tell you what I can. I wanted to move on to just introduce to people who are not used to working with scientists, how scientists in the non-commercial sector work. And I think this is a little bit odd when you're not used to it. Basically, after, I don't know, maybe 15 years of training, a scientist gets to become what's called a principal investigator, somebody who runs their own lab. And the best thing, from my point of view anyway, about being what's called a PI, not a private investigator, but a principal one, is that we get to choose the research questions that we work on. Nobody tells us what to do. We'll probably choose to work in an area where there are lots of scientists around us doing similar sorts of things, but not exactly the same or using the same tools as us. So for example, when I moved, I wanted to move somewhere where there are a whole bunch of frogs because I need frogs to do my work. But the research questions that my group asked were not similar to those around us. So we choose what research questions we work on. And then our main job in the first place is to persuade someone to fund that work. So you might have a great research question, but if nobody wants to fund it, it's not going to get done, not going to get answered. And those funding mechanisms vary between countries and institutions. Some countries have a core funding program, which is then supplemented by competitive grants, and some countries and some institutions have competitive grants only. And when I say competitive grants, I'm somebody who sits on a bunch of grant awarding panels across a number of countries, and they are very competitive. Perhaps 1 in 6 really good grant applications gets funded, maybe 1 in 5, but it's around that number. What does that pay for? It pays for people, and grants tend to last 3 or 5 years. So that means that you'll see people come and go from the Pura research program. And these are the people who are on these short-term grants. They also fund the consumables, some of the chemicals that we use, things like CRISPR, the Cas9 protein that makes CRISPR-Cas to do gene editing, are very expensive. And sometimes, but less often, it funds the equipment that tends to come in a different way. At the end, in the questions, if there's time, we could talk about how the fact that we are dependent on grants and how grants are very competitive can be an inhibitor to working, and remember the principal investigator chooses this, on working on an established rare genetic disease. And we could talk about that at the end, and I'm pretty sure some of the other scientists would have something to say about that as well. But I think it's an absolute credit to the Pura Syndrome Foundation that you still have, despite the fact that this is now an established genetic disease, a really quite significant research network that are still working on the basic biology or the clinically based research of this protein and gene. So this is, I think, largely due, or at least in large part due, to the enthusiasm and the communication that the group as a whole, including the patient network, have for this disease. So this is the group, and in red are people you'll hear from over the next little while. And there are changes from the last time that slide came up, and this is caused by the 3-year rotation of people on short-term grants. For some reason, my machine has stuck. Can everybody still see me? Am I, Damon, can you just come on and tell me if you can see me, or Eva?
You're here.
We can still see you, and your slides just started moving.
Thank you. Goodness me. There we go.
There you go.
Thank you. Sorry about that. So I wanted to start out by saying what the information was about Pura before it was known to be associated with a genetic disease. And this here is stolen from the Max Planck, a really nice diagram of a cell in its very simple form. In its simplest form, a cell has two really big compartments. One is called the nucleus and one is the cytosol. And in the nucleus is our DNA. And I've drawn a red blob there for where there's evidence or the possibility that Pura might function. And one of the first things that was known about Pura is that it would bind to DNA. And when it's bound to DNA, maybe it's involved in repairing damage to DNA. Maybe it's involved in controlling whether genes are switched on or off. So that's one possibility. When there's an mRNA in the nucleus, that mRNA had to be matured to start to be able to work and be exported from the nucleus. And it's possible, because we knew long before it was established that Pura protein was damaged in a disease, that it binds to mRNA. Out in the cytosol, because the mRNA goes out from the nucleus into the cytosol, well, there's mRNA metabolism. Various things happen to it out there, like its degradation can be controlled. Does the mRNA go on to do its actual job in the information flow, getting the information from DNA in the nucleus to becoming a protein? Proteins are, in the main, and everything I'm saying here, I have to say this because of my job, has a caveat on it, right? But protein is the main doing molecule in our cells. So it's the protein at the end of all this that we're really talking about here. So the mRNA might be degraded, or it might make protein. And the other thing that, because it binds to mRNA, Pura might do is control mRNA encoding protein. That's called translation. So it could control translation. So these are the places from our sort of prior knowledge of where Pura might work in that information to function pathway. Of course, we don't know everything about Pura. We didn't know everything about Pura. We certainly still don't. We could be missing something incredibly important, but these are the key places where it looks like it might work. Now, a few years ago, it would have been really difficult to start to understand the mechanism of Pura's action. Because actually, if we go back, if we can measure the mRNA in a cell and the protein in a cell and the mRNA in a nucleus, we can start to think about where Pura might work and start to work it out. And that has always seemed, to me at least, and I think to most of the discovery science researchers working on Pura, a really critical finding that will start to take us in the right direction to understanding the mechanism of how Pura works, and hence what's gone wrong when somebody has damaged the Pura protein. So we've got to discover the RNA changes and the protein changes that occur specifically in affected cells compared to normal cells, compared to controls. And we've got a number of models to do that, which we'll look at in a minute, from cultured cells, patient cells, model animals, and eventually, potentially, biobank samples may be very important in this, and particularly in personalizing the understanding of what's gone wrong in a particular individual. But the technology to do this has really come on enormously in the last few years. These are machines for analyzing RNA and DNA. This is one of the machines that analyzes protein. So nowadays we can analyze all at once all the different changes to RNAs that happen in a cell, all the different changes to proteins, and we can compare with and without Pura. And I'm going to come back to the first results from that right at the end, because that's the paper that is submitted for publication at the moment from Dirk's lab. But that's the exciting thing right now. the first clues might be coming about how Pura is working, what it's doing, what happens when you lose Pura from a cell. So what are our models for this? Here are our models. Obviously, human, not so much a model. Some days in my lab, we think humans are a good model for frogs, but that's probably wrong. Here they are. So we've got humans. Now, humans are obviously, in terms of what's most like a patient, that's a human. But there are problems with working with humans. Obviously, there are ethical ones and practical ones. And so much of the work that's done on humans is done in cultured cells. Now, these cells are, of course, human, but they're not able to talk to one another in the ways that they would in a whole animal. And of course, in terms of the human cells, we will also have biobank samples in the future that will be extremely valuable for some of this work. So that's our best model organism, a human, but it's limited by either simple cultured cells or even what are called organoids, where different types of cells are grown together. They seem to have limitations with rare human diseases at the moment, which I don't think we fully understand yet, but I'm sure they'll be overcome. So that will get ever more sophisticated and better. In evolutionary terms, our next closest neighbor is the mouse, and we're very fortunate working in Pura to have 2 mouse knockouts of the Pura gene. And you'll see in a minute that those are being used right now and the data being analyzed from experiments in them to try and understand what's going on when we knock out Pura in the mouse brain. And more recently, so since 2018, both in my lab here working on Xenopus, which is this particularly beautiful frog, I hope you'll agree, and in Tina Schmidt's lab working on zebrafish, we've got 2 other model organisms. Now, they're much less like humans, Xenopus more like humans than zebrafish, particularly genetically. But they have other advantages. Of course, an embryo within a mouse develops inside the mum, and there are very few of them, and they're very inaccessible. So it's very difficult to understand changes that happen in an embryo early in its development in mice. It's doable, but it's a bit of an epic battle, and it works much less well than in these 2 here, where you get hundreds and hundreds, or in the case of Xenopus, even thousands of eggs that develop outside the body and are accessible. So they let us work on very, very early development. And also they are very cheap and easy to genetically manipulate in the world of gene editing and CRISPR-Cas that I'm sure you've all heard of, than the mouse, which still stays, it's cheaper with CRISPR-Cas, but is still an expensive and relatively slow animal to manipulate genetically. So we've got we're fortunate, I think, in having everything from really good human cell models right down to all of the vertebrate models we would probably want to do our experiments. Zebrafish in particular may have an important role in the future, probably a long way away, but zebrafish can be used really well established for use in screening potential drugs to mitigate things. So we could envisage that a really robust Pura model in the zebrafish might conceivably be somewhere to look and see if things called orphan drugs could be used to mitigate any of the effects of Pura. So those are our model organisms and what sorts of things, it's stuck again, are we doing with them? Hurrah, we got there in the end. Okay, so the first one is to update you, I think, on the biobank. So this is being led by Dirk's lab, and there is already a huge biobank at the Helmholtz Centrum in Munich, and this is headed by Dr. Anton, loads of samples, and the Pura Foundation funds Simone to help set the biobank up, and this is incredibly valuable. And you can see that ethics approval has already been achieved. And I know from steering ethics committees that that's not a trivial thing at all. So that's a big thing. The preliminary website's been put together and there's the QR code for it down there. And then still ongoing is, but underway, setting up a biobank module at the Register, which is based in Southampton, about 15 miles along the coast from where I'm sitting. and also the getting together of the documents and instructions for running the Pura Biobank. There are some to-dos still. The long-term funding needs to be settled, and that should be this year. And then the launch of the biobank in spring next year with the full website. So that's going to be an incredibly valuable resource in the future in ways probably we can't yet predict, but ones that without doubt are going to be essential to understand Pura, especially at the individual level. So this is the paper that I'm going to tell you about right at the end, which is in a preprint form at the moment. So it's in what's called bioRxiv. And what it says is that depletion of the RNA binding Pura, so what Dirk and his colleagues have done is downregulate, take away a lot of the Pura from a normal human cell. So the standard, the sort of guinea pig, the laboratory guinea pig of the human cell world, HeLa cells. And then they've analyzed both the protein and the RNA changes that occur as a result. So that's exactly the sort of experiment that I was talking about earlier. Very exciting, really necessary to open the door to understanding Pura's function in human cells. So that's really exciting. We'll come back to it a little bit later. Also going on across the GRN, and surprisingly well coordinated given how busy we've all been in the time of COVID, is structural analysis of the human Pura protein and the impact of mutations. And that's being done biochemically in Dirk's lab, but also in a genetic way, just started by Sophie Powell in the Barale lab. And Sophie is a PhD student, jointly supervised by me as well. And she is funded by the British government through the BBSRC. And so what she's going to do is make mutations right across the Pura protein and see what their effect is in terms of cellular effects. So those two things together should help us start to understand if there are any links between a particular change in the Pura protein and some sort of change in the cell. So that's going to be important. And what we're hoping later on with Sophie is that we might extend, if we find some really important changes, we might extend that into an animal as well. Also in Dirk's lab is induced pluripotent stem cell research to understand Pura function from including patient cells, but in a more neural cell than the HeLa cells he's used in the paper preprint that's already out. In Dirk's lab as well, he's trying to understand in cells and biochemically the roles of Pura beta, sorry, I'm going to call them Pura B and Pura G in Pura function. And that again is complemented now by the fact that we have knockout Pura B and Pura G frogs in my lab made by two of our students, two of our master's students under the direction of Dr. Annie Godwin. So those pieces of data will start to join one another and help us understand the various roles of not just Pura alpha, Pura A, Pura, but also Pura B and Pura G. I'm really sorry about the sticky screen. Okay. So there are some other things going on that are very related to the work that I've just been telling you about in Dirk's lab with the HeLa cells. So from Jen Gordon's lab, brains were sent from control mice and mice with the Pura knockout. And in a team led by Diana Barale, those RNAs have been compared. And I was very excited to be in a meeting earlier this week where the first information from those is coming out, and we're starting to see what networks of genes, what networks of proteins in a brain are being affected when Pura is taken away. So again, this basic information that we need to really understand how Pura works. Similarly, we've got frog Pura knockout brains ready to compare with controls. again at the RNA level, sitting in our freezer, and that will be done shortly, we hope. And if we've got information from the cells, the human cells, from mice and from frogs, if we see the same networks or some of the same networks are affected in all of them, because they're affected across evolution, we know from lots of other studies that these are most likely to be the real key changes. And this will highlight from a very complicated group of data what the most likely important changes as a result of losing Pura are. So we're very hopeful that this is going to move forward relatively quickly now. The material is there, the analysis needs to be done in some cases. So I wanted to go back to where might Pura work. And this is really some of the information that's easy to extract for you today from the preprint from Dirk's lab. And what they found, and you'll see much more about this really interesting finding in a minute, is that mainly Pura is not in the nucleus. So it's unlikely, but not impossible, because you only need tiny amounts of a protein to do these things, that it's involved in these. Also, that it's not, there's no change when you take Pura away of the match between mRNA and protein levels. So it doesn't look like it's working here. But what there is a change in the amount of nearly 4,000 different mRNAs. So it's something here, something to do with the way the RNA is treated in the cytoplasm almost certainly, although we can't absolutely exclude something in the nucleus, I guess, but it's something out here that's important. And that's the first sort of piece of information as to where we might be needing to look to understand where Pura is working. So that's a very exciting result for me, but I wanted to quote the last words of that preprint paper. While this study opened the door for an unbiased and systematic understanding of molecular Pura-dependent pathways, it will require more research on both ends, clinical and non-clinical, to convincingly connect our findings with the symptoms in Pura syndrome patients. So even though we're starting to understand the mechanism, there's still a lot of work to do. This is the group. This is the group that's been really supportive of me in particular as I've taken this on. Dirk, Tina, and Di, and thank you so much for your help. And this is kind of where we are. The clinical data, the animal models, the cell biology, and the structural biochemistry are all starting to produce a picture of something around the molecular work of Pura in the cell. So we're part way to understanding how Pura works. And then we can start to look more closely at these possibilities that could lead to a therapy sometime in the future. Thank you so much for your attention. Please do talk to us. Please ask questions. I'm certainly very happy to answer them, and I'm hoping I will be here next Saturday as well to answer them then. I shall stop sharing my screen, and if anyone has any questions, they're very welcome.
Thank you, Dr. Gill. That was very informative. We do have some questions rolling in, I believe. The first one is from Eva Kate. Could you explain more about Pura B and Pura G, what they are and their relation to Pura? Okay,
so there are three very similar proteins in the cell. To Pura A and Pura, I'm sure you're really familiar with. Pura B and Pura G are also similar. We know that they can interact physically with one another in the cell. What we know much less about at the moment is what do Pura B and particularly Pura G do. Pura B we know is associated with a particular leukemia, and it seems to be important in cardiovascular development, I think would be fair to say, from other models, but also our own findings, that there's a strong hint that it's important in that. To what extent they overlap in function with Pura, I think is a really good question, and not one that we have a firm answer to yet, in my understanding.
Okay, another question might be similar. Can you share what are the phenotypes of pure A, pure B, and pure G? And I
frogs.
think in frogs.
Yeah. So, oh my goodness, I'm not the very best person to answer this, but I'll give it a good go. So, in Pura, if we make a complete knockout, then they are quite badly deformed craniofacially, the tadpoles. And if we make a really complete knockout, they struggle to survive. When we make heterozygous, so that's one of the two genes being knocked out, then they survive a lot better. And we're looking at the moment about what the actual phenotype is. So that's the model, like the human disease, where one pure gene is normal, one is damaged. Pure B and pure G, we only have total knockouts at the moment. Pure B is a very poorly frog, and they don't survive well at all. Pure G looks pretty normal. We're just doing behavioral analysis on those tadpoles right now, because pure G in the tadpole seems to be expressed only in the brain, whereas pure B is expressed elsewhere as well. So pure B, yeah, they're not well, and they're not well in multiple ways. And we still haven't finished working out what all those ways are yet. Pure G, they seem normal, which we didn't expect. So we haven't found what's wrong with those yet. They seem to be okay.
Another question: if the change is not in the nucleus of the DNA, does that mean it is not transmittal through hereditary?
No, it's still hereditary. So the change is caused by, so initially there's sort of a pathway here, isn't there? The DNA is damaged. So part of the, so the Pura protein that's made is damaged. So if you then have offspring from a person with a damaged Pura gene, their offspring will have a certain chance of having a damaged Pura gene. The fact that the Pura protein works out probably in the cytosol of the cell, not in the nucleus, that's a different fact, okay? That's in a different space. That's a different piece of knowledge, rather than the DNA is still always going to be damaged. Even though the protein's working out here, the DNA is damaged. So unfortunately, no, it doesn't mean that it's not hereditary. It's still hereditary.
Okay, I don't see any more questions in the chat. So thank you, Dr. Gill.
Thank you very much indeed.
And if there are any questions that you think of later, you can always go to the website and we'll answer it on the panel.
Brilliant. Thank you very much, Dan.
Thank you, sir.